DNA is made up of nucleotides that are bonded together. These nucleotides include a nitrogenous base, a phosphate group, and a deoxyribose sugar. The nitrogenous bases are paired together. Adenine and thymine always pair together, and cytosine and guanine always pair together. These bases are bonded by hydrogen bonds. DNA is a double helix. If you untwisted the helix, a DNA ladder would be present. DNA is antiparallel. This means the opposite sides of the strands are connected. Therefore, the longer head of one strand is with the tail of the other. Transcription is the process of DNA being copied into RNA. The language stays the same in nucleotides. It happens in the nucleus of a cell. First, RNA polymerase splits the double helix and starts forming messenger RNA in a complementary way. Guanine in the DNA becomes cytosine in the RNA. Cytosine becomes guanine. Thymine becomes adenine, but adenine becomes a new nitrogenous base called uracil. Promoters signal to the polymerase where it should start making RNA. Promoters have specific base sequences that code for specific proteins. After this copying process occurs, the DNA zips back up and the RNA transcript is ready to be edited. RNA editing takes place after transcriptions. The portions of the pre-messenger RNA that are cut out and discarded are called introns, while the parts that are kept and put back together are called exons. A cap is then put on to the beginning of the RNA. Then a poly A tail is put on to the end.
Translation is the process by which the genetic code is translated into a chain of amino acids called the polypeptide chain. A strand of messenger RNA leaves the nucleus of the cell and passes through a ribosome. As it passes through, each codon, which is 3 nucleotides, is translated into a single amino acid through transfer RNA. Transfer RNA brings the complementary amino acid to the ribosome. The codons of messenger RNA correspond with the anticodons of transfer RNA. When the amino acid breaks off the anticodon leaves to go find another ribosome.
Amino acid chains fold into proteins in 4 levels. The first level is the primary structure. This is a polypeptide chain. This is a sequence of a chain of amino acids. The second level is a secondary structure. This is hydrogen bonding of the peptide backbone, and it causes the amino acids to fold into a repeating pattern like an alpha helix or a pleated sheet. The third level is the tertiary structure. This is a 3D folding pattern of a protein due to side chain interactions. This is also the minimum level of an enzyme because it is irregular and has an active site for the substrates to fit into. The fourth level is the quaternary structure. This is a protein consisting of more than one amino acid chain.
When we hear scientists say that proteins make you, they're not wrong. Proteins create enzymes which act as catalysts, speeding up chemical reactions in living things. These chemical reactions are necessary to the function and health of almost all organisms.